Hi, I'm Grace. I work at the Biomedical Imaging Unit in Southampton, and part of my job here is to process the samples for electron microscopy.
So I'm going to talk to you about tissue processing and all the things that it involves. So firstly, I'll start by saying that we receive specimens in the form of biopsies, and my job is to process them into a block that can be cut and then looked at on the electron microscope. So there's lots of stages to tissue processing. It's a very structured process with lots of timings, and I'll just talk you through the basics. So the first thing we've got to do is preserve the tissue. So straight away when the biopsy is taken, the tissue is put straight into glutaraldehyde. So glutaraldehyde is a fixative, and what it's going to do, it's going to instantly preserve the structure of the tissue. After the fixation process, the tissue needs to be rinsed in a chemical buffer to remove the excess molecules of fixative. This buffering process also stabilises and maintains the pH of our specimen. The buffer that we use is called sodium cocodylate. It's made from a poisonous chemical and it contains arsenic. So now we're going to move on to the osmium stage. Osmium is the densest of all natural elements, and in the world of microscopy it's made into osmium tetroxide and it's used as a stain and a secondary fixative. So osmium tetroxide reacts to lipid molecules in the tissue, and so when the osmium targets these areas it will add density to all of the lipid structures. The membranes of the cell are also composed of lipids. It's a phospholipid bilayer, so this means that the whole membrane gets stained by osmium. So staining is a really essential part of the processing. I've just put two pictures here, one with osmium and one without, just to show you the difference. You can quite clearly see the one on the left that has been stained. You can really define the structures. There's a really nice contrast between the light and dark areas compared to the one on the right, which has not been stained. You can see that the whole thing is just very pale and lacking contrast. It's quite hard to see what's going on in comparison. This time-lapse video I think shows really nicely how osmium works and how it completely changes the colour of the specimen. Uranyl acetate is a bright yellow compound derived from uranium, and in microscopy it's used as a stain. Uranyl acetate targets the proteins and nucleic acids, so this is going to affect the ribosomes and the nucleus. The reason for the staining is going to improve microscopic quality because it's going to add contrast to the targeted areas. So we use ethanol to gradually dehydrate the specimens. So we start with a concentration of 70% ethanol and gradually increase that until we get to 100% ethanol. The aim of this is to chemically extract any remaining water, and this is so that the specimens can be put under vacuum. Acetonitrile is a type of solvent. We use it after the ethanol stage as a link reagent to resin. Acetonitrile was used on its own to start with, followed by a 50-50 mix with resin. The acetonitrile helps the resin molecules infiltrate the specimen before the complete transition to resin. We use epoxy resin to form the block that the specimen is embedded in. At room temperature, the resin will be a viscous liquid that can be poured and pipetted, but when heated to 60 degrees in a process called polymerization, the resin solidifies into a block. So I'm just going to leave the specimen in fresh resin now for 6 hours, and this will ensure that the specimen is fully infiltrated by the resin. So we use these plastic capsules to form the mould for our resin blocks. So I'm going to transfer the tissue into the bottom of the capsule and then I will fill it with resin. And it's always good practice to adjust the specimen so that it's placed centrally. Once we've done that, the specimen is ready to be polymerized. So I'm going to leave it in the oven overnight. We'll now fast forward to the next day, and as you can see, the resin has completely transformed into a solid structure. I'm going to carefully slice the capsule open using a blade to retrieve the block. So here is the finished result, and that means that the processing is now complete. So you can now follow the next stage of the specimen journey, which is microtomy. You're going to learn all about it, but this is where the block is now going to be cut to produce microscopic sections. I hope you enjoyed this video and thank you so much for watching.